Hello everyone, we are excited to present our research project titled Exploring the Role of Antisense Oligonucleotides in Regulating Non-Coding RNAs in Salmonella Typhi using Cationic Liposomes. Before we begin, let us meet our team where the presenters will be Mahesh Ramesh and myself, Darshan Kumar, and the other team members include Dr. Sumita Samugam, Associate Professor Dr. Venkata Suresh Chini, and also Associate Professor Dr. Vijayan Venugopal.
This project is part of our effort to develop novel RNA-based strategies to weaken multidrug-resistant strains of this pathogen. Typhoid fever continues to be a major global health problem, affecting more than 11 to 18 million people each year and causing over 100,000 deaths. The highest burdens is seen in South Asia, which accounts for almost 70% of global cases. These numbers highlight the urgent need for new and effective treatment strategies. Unfortunately, the standard antibiotics are failing against multidrug-resistant and extensively drug-resistant Salmonella Typhi. Drugs like ampicillin, chloramphenicol, and ciprofloxacin are becoming ineffective, and even ceftriaxone and azithromycin-resistant strains are emerging. This growing resistance crisis threatens to make typhoid fever much harder to treat.
To tackle this challenge, our research focuses on the SPI7 pathogenicity island, a region in Salmonella Typhi that carries important virulence genes. In SPI7, there are many non-coding RNAs, but there are three non-coding RNAs that are identified which can regulate genes critical for infection, which are STYR9, STYR143, and STYR381. Our approach is to design antisense oligonucleotides, shown here in green, that specifically bind to and silence these ncRNAs by switching off these regulatory RNAs. We aim to weaken the bacterium's ability to cause disease, opening the door to a new RNA-based therapeutic strategy.
To test this idea either working or not, we first design and synthesize antisense oligonucleotides, which is targeting the three main ncRNAs.
Next, we encapsulated the antisense oligonucleotides in cationic liposomes to improve delivery efficiency and protect them from degradation. These liposomes facilitate uptake into Salmonella Typhi cells, allowing the antisense oligonucleotides to reach their target RNAs.
We also then perform a growth and a killing assays to measure the bacterial survival after treatment and detect how much the bacterial survivals had grown. Dot blot assays were used to confirm that our ASO specifically bind their target RNAs, and it is allowed for fast and convenient detections and identification of nucleic acids and proteins too. Finally, the RNA sequencing and pathway analysis allow us to examine global gene expression changes and identify disrupted virulence pathways that distract the gene expression pathways.
Our results show that antisense oligonucleotide treatment significantly reduced bacterial growth as seen in the survival curves. We also observed a marked decrease in the expression of SPI7 regulated virulence genes, which can be justified via bioinformatic or pathway analysis as well, and it confirms that our antisense oligonucleotides successfully silence their RNA targets. Our approach shows that targeting ncRNAs with antisense oligonucleotides can effectively suppress virulence in Salmonella Typhi. This means fewer infections, better treatment outcomes, and ultimately healthier communities protected from the burden of multidrug-resistant typhoids. Together, these findings support the potential of RNA-based therapies as a novel approach to control multidrug-resistant Salmonella Typhi and reduce disease severity. On behalf of our entire team, we would like to sincerely thank everyone who watched this video and supported our project. We extend our gratitude to AIMS University for their unwavering support. And finally, we want to thank MJIC for organizing this virtual video competition and giving us the opportunity to share our work with a wider audience.